And I want to now welcome our moderator for this evening. We want to welcome Robin Ka'ai. He is a former Kapiolani Community College alumni, and now he's a graduate researcher at Hutch United. So let's all welcome Robin.
Hi, everyone. Good evening. I guess good afternoon to people in Hawaii. Good evening to anyone joining us from Washington. It feels very late. It's dark outside. And so I'll be moderating the rest of the career and science panels. I know we went over some housekeeping. I just want to reiterate, feel free to turn on your cameras if you want to, but we just ask that you mute your microphones during at least the panelist section. We will have a question and answer segment where you can directly interact with them. So if you have any like real burning questions, please put them in the chat so we have a chance to look them over and give them out to some of the panelists so they can think about it as we're approaching that Q&A section. And lastly, that the link to the feedback survey and the sign-in, it's very important to us. We want to know what you think about this panel so that we can hopefully do this in the future. Things that you like, things you didn't like, what you want to see more of, or any suggestions for how we could do this better, especially in this very unprecedented time. Please let us know. We'd love to know so that we can do more of this for the students. And I guess with that, we can get rolling with the panel. I'd like to start off with, publicly saying, thank you so much to our panelists for showing up and making time in your very busy schedules to join this webinar and talk to the students and share your journey through science with us. We have a very amazing assembly of panelists lined up, so I'm pretty excited about that. And I also want to thank, Leanne and Calais for helping with the coordination efforts since this is across the Pacific. And just helping me connect these professionals with the students that are there at KCC right now. So I'm very thankful for that. And the reason for this panel is that when I was an undergraduate in the University of Hawaii system, I didn't have a lot of exposure to job prospects as a biological science major in Hawaii. It was more or less limited to you could be a researcher in an academic institution as a professor, which those positions are very hard to come by, or you went to med school and became a doctor instead. And so this was kind of my answer to past Robin's conundrum with what do I do if I major in microbiology or molecular cell bio in a place where we don't really have a biotech industry or very large advertisements for what you can do at a hospital with your degree or teaching without having to do nothing but intensive research. So this is something I want to give back to KCC for everything that they've given me over the years. And a little background on that is that when I first got to KCC, that's when I was introduced to that science component in education and what I wanted to do in the future. They really helped me by providing stipends and undergraduate research. They introduced me to Matthew Tuthill, or I guess the reverse. Matthew Tuthill was my first PI. Introduced me to the KCC STEM program, and from there, that's where I learned outreach to local high schools for summer programs. So it was a great program, and this is another way that I kind of want to give back to that, especially where I am right now, which is at the Fred Hutchinson Cancer Research Center. Still pursuing my PhD, I still reach out to KCC for things like letters of recommendation or advice on what I might or might not do as I progress through my own academic journey. But that's who I am and the reason why this panel is existing right now. And I guess to talk more about KCC and the other things that they offer, since I'm sort of I feel out of tune because I haven't been at KCC for so long. My old PI, Matthew Tuthill, is here. He's just gonna give a quick pitch as to what KCC has to offer some of the students that might attend there in the future.
Yeah, I just want to thank you guys for giving me an opportunity to speak for about a minute or two. And I want to thank Robin and the panelists for putting this together. I'm excited to learn from you guys, and I think a lot of us are as well. And then I want to say to Robin, you know, physically you might be far away, Robin, but I think your heart still belongs on this campus, or at least parts of it seem to be. Yeah. In terms of biotechnology that we offer at our community college, as Robin alluded to, we're really unique because we actually offer tissue culture, we do antibody production and characterization, and recombinant DNA work, all at the community college level. Because experience tells us that as challenging as our projects may be, if they're presented properly, the students really excel at it, and they actually move really fast through the material. Robin's also a really good example of that. I would ask some of the students who are at Kapiolani campus to consider some of the opportunities that are available to you that open up the doorway for all sorts of biomedical related fields. Many of us are very narrow in the paths that we're going, but I would ask that you talk to some of your fellow students and talk to some of the instructors on this campus and all across the STEM pipeline, and realize that there are a lot of opportunities. You don't just have to focus on what's been presented before you. Robin, I think, discovered that. Wouldn't you say, Robin? You had a path set, but over time that really evolved into something different.
Oh yeah, I mean, this is how most of these panelists got here in one form or another. You know, experiences kind of tripping through science had led me to things like right after, you know, I finished my undergraduate degree in 2015 at the University of Hawaii. I had in a molecular cell bio degree, and I didn't really know what to do with it, so I just went straight to work at you know, Straub Medical Center, which is where I met Ramelin. And that's where I learned, you know, diagnostic medicine and seeing the direct impact of, you know, our advances in science and technology and how that, you know, improved patient outcomes and stuff like that. And then I was offered a position with, you know, the state working on whole genome sequencing and using a more epidemiological approach to help the community. And that's where I, you know, met Samantha. And with Tracy Delgado and Jillian, I was actually tipped off by people at the Hutch, where I do research now, to just cold call them. And being who I am, I just started sending out emails like, they can say no, definitely, but I will give them that chance by emailing them out of the blue. And so it all started, I think, with KCC. I was originally a nursing major, and I got into nursing school at the University of Hawaii, but it was when I took microbiology that I realized that, you know, if I could get someone to pay me to do this, that would be an amazing career opportunity. I still, every now and then, I look back at like, I could have just been a nurse and made this much money and not had to worry about why my experiments are failing all the time. But I think I'm still happy with the decision that I made up to this point about, you know, diving in and finding these very specific problems that you can solve with science. And that's why, you know, I collected all these people that sort of represent the aspects of what I kind of went through and all these opportunities that, you know, were presented before me. So that, you know, students at KCC, or students, because we still have some people attending from Washington, you can see that, you know, you don't have to decide on one particular job right this moment. You can start journeying through science, and there are so many branching off points that you can go through, because I went through a bunch of them, and now I'm at the University of Washington, where I never really thought I'd be. But I want you guys to hear their stories. They're amazing stories. These are amazing people. And hopefully this inspires someone to be like, oh yeah, I never really thought of being an educator, you know, as a researcher, or doing something in industry, which is, you know, difficult for people in Hawaii because we don't have industry. And so that's why I really wanted to reach out to like Jillian, or the fact that you can take, you know, what you learn in biology and apply it to something that you had no idea you would. So yeah, I think KCC offered me, you know, a really good jump off point to sample all these different pathways. And so I just want to bring them together so other people can see what you can do with a biological science degree, because I don't think it's advertised enough. Did you have anything else you wanted to talk about with the program, Matt?
No, I'm good. I actually want to learn about other things from other folks. I'm excited.
Yeah,
Thank you.
I know, me too. I'm so tired of hearing myself talk after all the Zoom meetings I went through today. I don't understand how educators do this. I come from a family of educators. Ashley, my girlfriend, comes from a family of educators. I don't understand how you have the endurance to do this every day. But I guess just to introduce our panel of people right now, I'll just give you their names, of like a brief background on them, and then we'll get into our first speaker. From the Hawaii State Department of Health State Laboratory Division, we have Samantha Silvis. She's over there. If you could wave hi, Samantha. She's actually at work right now. So those are the blue scrubs I used to wear Monday through Friday. Oh man, I miss being able to just not worry about what I wore to work. We also have Ramlin Gonzalez. She is a med lab tech. Ramlin, if you could wave for us. She just woke up because she's typically on night shift. So I dragged her out of that circadian rhythm so that she could come talk to you about the crazy world of being an MLT in a hospital. She works downtown Honolulu in Stroud Medical Center. That place is like, it gets pretty high speed and high stress, but if anyone is interested in that kind of environment of high-paced learning and working, I mean, it's definitely a career option. We have Jillian Bruffy. She's, there we go. She is from the Protein Design Institute at the University of Washington. She was one of my cold calls. I know her through Hannah Lewis, who told me to reach out to her. She does work with protein design in the Baker Lab. Last we have Dr. Tracy Delgado, who is a associate professor of biology at Seattle Pacific University. If you just give us a wave, Tracy. She had an amazing TED Talk. She does a lot of work with undergraduate students and getting them into science with a really, really great focus on education, which is, that hits home for me because of, what my family does and what Ashley, my girlfriend's family does. I don't want to do it because of watching my mom go through all these report cards and teaching assessments and listening to Department of Education and all that stuff. It's crazy, and I appreciate what you do, but I don't know how you how the educators get through all that. So I'm glad all of you are here today. I think today we're going to start off with Samantha talking to us about what her job entails at the state of Hawaii and what she does on a day-to-day basis, especially now in these very odd pandemic times. Samantha, would you like to unmute for us? Hi, how's it going?
Good, busy.
So tell us a little bit about how you ended up doing what you do and what you actually do, and take us through a day in the life of Samantha.
Okay. So I started off, I went to the university at Stony Brook University out on Long Island. I have my bachelor's of engineering in biomedical engineering. I enrolled in graduate school at the University of Buffalo, all gung-ho that I'm going into cancer research with the idea that I was going to get into developing cancer drugs on a pharmaceutical level, working in industry rather than working, in academia. About 6 years into my project, things fell apart. We hit a wall, lab lost funding, and I was put between essentially a rock and a hard place of finding a brand new lab and starting over, or just step away and take my master's. And I chose the second. So I left the University of Buffalo. I took my master's in essentially molecular pharmacology, moved out to Hawaii, and was left with a bit of a conundrum. What do I do in Hawaii when all of my education is from a pharmaceutical background? There's really isn't much option for me out here. I found a position working at the Department of Health at the state laboratory, just as a lab assistant, just trying to find something, get my foot in the door, just a very basic entry level thing. What happened was about maybe like a couple of months into my new job as a lab assistant, Hawaii was hit with a mumps outbreak, and the bioterrorism department, who was doing all of the testing, kind of overwhelmed. So I had way too much free time on my hand, and I started volunteering and helping them out with getting the specimens in, accessioning them, making all of the data entry checks, getting all of that done. And they started bringing me in and training me on different things. And then when a contract position opened up with them, I jumped at it and I started moving into doing their arbovirus testing. So I was testing for dengue, I was testing for Zika, I was testing for chickpea, any of those mosquito-borne illnesses that are very prevalent in Southeast Asia, in the Pacific Islands. A lot of tourists come back and they have it. And, we're trying to avoid dengue outbreaks here in Hawaii, which have been a problem in the past. So I started that testing. And then about a year and a half down the line, another job opened up within the same department doing something completely different. And this was definitely a more microbiology-based career move for me. And I have never taken a microbiology class as part of like a college curriculum. So it's definitely me moving into a new area. And what my job currently entails is I work as part of a laboratory response network, testing for any type of agents that could be used in like a biologic attack. Anytime there's a mysterious white powder that gets found that they're concerned it might be ricin, it might be anthrax, that sample comes to me and I'm the one who does the testing to determine that. But everything's quiet on that front right now, and we are all consumed doing COVID testing. So my life for the past, I don't even know how many months now, has all been COVID. We are very, very busy up here, and we're always welcoming more microbiologists. We always have, we have contract positions opening up, especially now, because we need all of this extra help. And yeah, so that's pretty much what I do over here at the Department of Health. Robin, do you have anything?
How, I guess, I feel like I can't ask certain things because you're at work right now. How do you enjoy working in a governmental lab as compared to a research lab in academia?
Yeah.
How is that?
It's definitely a big change where research, it's more free flowing for research, and working on a state job, it's more regulated, but it's also still free flowing in some areas. Like I come into work some days and I don't know what I'm going to have to do today. It depends on what samples come in. My day, I can have everything all planned out at the start of the day, and then something high priority comes in and I have to drop everything and focus my attention on that. My day and if something high priority comes in like that, my day might not end until 8 o'clock at night, 10 o'clock. I've been here since, until 2 in the morning before. We come in on weekends, whatever the testing demands. And it all revolves around that, because what we're doing, the tests that we're doing, you can't lose sight of that patient sample is attached to somebody who's worried, somebody whose family's worried, somebody who's not well, and I'm able to put my time and energy into giving them answers, even if it's just something as simple as do I have COVID or not? I find it rewarding that I can indirectly give somebody, a little bit of peace of mind, or, give them answers to questions that they don't have the ability to know themselves.
And how much of your work is affected by the inner workings of the rest of the government? Like when the government has a decree, or if someone else in the Department of Health is like having some issue, how much of that is actually, or I guess how involved are you? Because most of the people I think have this idea of the archetypical laboratorian as someone that's just like in this enclosed space, they do their work and like they're in this isolated area. How true is that as far as a governmental lab? Like, how do you feel?
We have a little bit of that because we are kind of isolated away from some of the people who make the big decisions, but we also can have our own input. Like a lot of the grants for our funding for our job positions get written by the people who are in that area doing that test. And a lot of it's joint efforts between us, the laboratorians, and downtown in Honolulu, where the EPIs are, the ones who are gathering all of the data on our state as a whole population. Like back in, I think it was 2015, we had the dengue outbreak on the Big Island. So we have to constantly monitor all of those kind of arbovirus, mosquito-borne illnesses within our community. So once we have the EPIs collect all that data and we are able to do the testing here, we can apply for grant funding and, you know, from the federal government. So we can have like our own safety net to better care for our people in our state.
No, that's great. No, thank you. I know I have very jaded answers in my head for some of these questions that I'm asking you. It's like, oh, let's get it to a meeting with department heads from places that have nothing to do with us and argue about PPE, because that seems to be what we really enjoy doing. But I mean, I guess that can be, you can say that about any institution. I think it's just more pronounced in governmental work. Okay, so yeah, unfortunately, some of the people here are still working, especially with Samantha. We were lucky to kind of catch her midday when she's on a sort of lunch break, and then I just convinced her to come onto this call. Dr. Tracy Delgado, as I said before, is the associate professor of biology at Seattle University. So, Tracy, can you hear me?
Yeah, I can hear you. Can you hear me?
Yeah. So, if you could, you have again an amazing career. I know that you like to champion. I guess like underserved and underrepresented peoples in science, because I'm an active SOCNIS member, and you know, we tried to invite some of the people from our chapter there. Could you tell us about, you know, your journey in science and where you ended up and what you kind of enjoy about where you're at?
Yeah, sure. So I'll give you like the 10-minute or less version of that story. And so I actually went to UCLA. I didn't go to community college, but I definitely had lack of preparation for college, I would say. So I came into UCLA, you know, honor student, ready to kind of, you know, be the best as I could be. And then I get there and realized it was really hard. And due to lack of preparation, I come from East LA. And so, you know, it wasn't the greatest educational system. And so it definitely was a struggle. You know, I was getting C's and I was a microbiology, immunology, and molecular genetics major. It's all one major, even though it's all like sounds complicated. That's how UCLA worked, maybe to make it sound fancier. I don't know. But so I was majoring, and I was like, I really wanted to be a scientist, but then I thought like, man, there's no way, you know, I could do this with these grades. And one of the key things that really helped me become successful was to surround myself with peers and other people who come from similar backgrounds or similar struggles to support one another. And that pretty much made the difference for me to be successful in college. And so those C's became B's. And by the time I got junior, senior year, those B's became A's. And I was really excited to, you know, continue on and go on to graduate school. And I spent a lot of time doing research while as an undergraduate, because I knew I wanted to go to graduate school and that was going to be important. And that's really probably what saved me, because I had a lower GPA, being able to do research all 4 years of undergrad. I started as a freshman, like I was just going in, I was like, let me get in here and, you know, learn how to be a scientist. So, and doing summer programs and stuff. And so I got to the University of Washington and decided to major in, or get my PhD in microbiology. I really was fascinated with viruses. As a undergrad, I learned about viruses, and it's like, they're not even alive, but they cause such crazy diseases, which is, you know, out of this world. And as we know, we're in a pandemic that is caused by a virus. So you can probably appreciate virologists now and what they're doing. And so I was really interested in viruses, but I also had a strong history with doing research with cancer. And so, you know, I really like that kind of science as well. And it's kind of more of a cell biology, cancer biology field. And so I went to University of Washington and thought, well, let me see if I can find a way to do viruses, but maybe also do cancer as well. And I found a lab that studied how herpes viruses cause cancer, because about 15% of cancers are caused by viruses. And so we were just like, well, you know, let's let's go ahead and do that. And I found a great lab, and kind of again suffered from imposter syndrome, and how am I gonna do this, and especially being a person of color coming in and not having community, it was gonna be really hard for me. But luckily, I got involved with SACNAS, which is the Society for the Advancement of Chicanos and Native Americans in Sciences. It's a national society, but they also have local chapters that can be implemented in schools and universities and even community colleges as well. And so we made a chapter and found like my people to help me again, provide that mentorship and support needed to kind of get through grad school. And so even though, I struggled at least in the beginning with just finding that community, by the end, it was going really well, and was able to get my PhD 5 years later in microbiology. And through that aspect, I also was figuring out, like, I always kind of wanted to be a PI or like a professor, but I didn't know to what extent I wanted to be like the research professor who works at the big school and that's all they kind of do. And teaching is like kind of a chore than something you want to do in some aspects. And I TA'd and I loved teaching. Like I loved seeing, the students and learning and these little light bulbs go on. And so when I was in graduate school, I started to teach. Like I taught like maybe my 4th year, one summer, I taught like a microbiology class at a community college. And I said, this is a great opportunity to teach and be the professor and teach microbiology and see what that's like and see if this is what I want to do. And I did that and it was great and I loved it. And I said, this is what I want to do. I kind of want to teach. I still kind of want to do research, but trying to figure out that balance. I networked with a bunch of deans who are like the bosses in academia and said, hey, I'm graduating soon. If there's a job, let me know. And mostly just said, sorry, we don't have any openings yet. We'll let you know. And then a couple months before I graduated, one university was like, hey, we actually have a position, you should apply. And I applied and got the job, which was amazing. And it was at a university that I wasn't at right now. It's Northwest University by Seattle. And that's a purely mainly teaching, no research aspect. And so I went there and I was teaching, full load and loved it. Of course, just being able to, feed the next generation the science of the future, which is great. But then I started really missing the research aspect again of doing the science. And that was about the time I did the TED Talk, which basically describes how we may be able to use metabolism inhibitor drugs that may be used for cancer, using them against viruses. And so it was kind of a new idea that was kind of, well, not new, but newish idea at the time of, this is something we should be thinking about. And I got really intrigued and really excited. And I said, I want to start my lab, but no one's doing research here. So I'm just gonna do it by myself, which is kind of, when you're the only one doing research, it's a big endeavor, getting the school set up, building a tissue culture, biosafety level 2 herpes virus lab, plus molecular biology space. And I train undergrads. And so my undergrads are the ones who got to do all the experiments. My undergrads are now in like med schools and grad schools and won awards at conferences for their research, and things went really well. And then I felt lonely again because I felt like I was the only one doing research. And it was like, because the expectation was not to do it, right? But I did it be above and beyond because I wanted to. So I decided to switch universities for the sake of having more of a balance between scholarship, research, and teaching. Mm-hmm. And so I recently moved to Seattle Pacific University pre-pandemic last fall. And it was a great move because I felt like everybody does research, it's expected, everybody publishes as part of tenure and part of promotion, but everybody also gets to teach courses, not as heavy of a load as I did last university, but still a decent teaching load. And I've loved it. I loved training students both in the classroom as well as in the lab. I like to think of it like a ripple effect, where whatever I plant in them, it's like the first ripple when you have like throw something into the water, and then they use that knowledge and disseminate that to the people around them, and then that disseminates from there. So it's like, well, maybe I'm not out there right now doing COVID stuff, but I've trained students who now have gone on and are doing COVID stuff. So it's like, you kind of like that's a very rewarding experience. And then like you were saying, Robin, you know, sometimes your experiments don't work. I see teaching like a constant satisfaction, and then when research is like, meh, like it's not going so well, and then you get like the big high, and it like supposed to last you for a while, and who knows how long that's going to be before you get a high again. So I feel like with my teacher scholar like position, I get like my constant satisfaction with research. I mean, not with teaching, and then I get to do like my little like ups and downs with research. So it kind of provides best of both worlds.
Okay, no, that's a great thing to speak to, and I think it's very important to impress on people that, you know, even in research, this whole idea of mentorship and education, it's still there. You still have to text, you have to teach stuff, and if you're unwilling to do that, it's like you're gonna have a very lonely, difficult time doing all these experiments by yourself. Maybe that's something you want, but definitely mentorship does play a massive role. And I guess as far as, you know, the viruses causing cancer, I work in an adenovirus lab, and you know, first known virus to cause cancer. Shout out to adenovirus. But thank you so much for sharing with us, Tracy. We're gonna move on to the next speaker for time's sake. But yeah, hopefully we can come back and ask you more questions about scholarship and also education. So our next speaker is Jillian Bruffy. She is, like I said before, a graduate student with the Baker Lab in the Protein Design Institute. And Jillian, I know that you previously worked in the industry, and that was one of the reasons that it attracted me to call you and ask you whether or not you wanted to speak on this panel, because we don't have industry in Hawaii, and so I thought it was like a very valuable thing to kind of impress on the students that that is a viable career, maybe not in Hawaii yet until we build the infrastructure. But could you tell us about some of your experience, like leading up to getting up to where you are and where you kind of want to go after you leave the Protein Design Institute, if you do?
Yeah, for sure. Thanks for the intro. So, hey everyone. As Robin said, I previously to grad school worked in industry, and my plans for after grad school are to go back to working in industry. And kind of the path that took me to this point is I graduated undergrad with a degree in bio and chemistry. I kind of had the wrong mentality as an undergrad and didn't realize that getting lab experience while I was there was way more important than like completely overloading my class schedule. So I missed that memo that Tracy had. So when I graduated, I had like 1 summer's worth of experience during protein purification. And fortunately, that was enough to get me into a nice internship right after I graduated. So it was a little bit scary going out into the world. Like I finished my degree and I just have an internship, not a full job. But that at least I had an internship at Genentech where I worked for that summer doing more protein purification there. And thankfully through that, I was able to morph that into a temp job, also there doing more and more protein purification, which I highly recommend as like an intro job. Looking into temp jobs, it's a really great way to get your foot in the door, get more experience, and make some connections, because unfortunately in industry, connections are huge. So from there, once that temp job was up, I found a job at a small like 20-person biotech company called Nurix, doing drug development. So I was more purifying proteins now to like figure out assays to look for small molecule drugs that could block the activity of these proteins and then could be used as like an anti-cancer agent. So I really loved the work there. I loved that I was doing something that was so proximal to like making something to go out into the world and help people that will hopefully at some point be a real treatment. So I really loved that. But while I was in industry, I kind of came to this revelation that industry is very split into 2 career paths, the one for people without PhDs and one for people with PhDs. And if I wanted to move into that other career path where you get to do more of the like deciding what projects people work on and doing like kind of the background research of like, why are we pursuing this target? Why is it cool? And like leading a group of some sort, I would need to go back into school and go back and get a PhD. So it was hard to leave industry because honestly, I still like it a lot more than I like the academic lab workflow. But I decided that it was really important for what my career goals were to go back into school. So from there, I applied to the University of Washington where I, as Robin said, joined David Baker's group in the Institute for Protein Design, where there I work on, as the name states, designing proteins. I kind of kept my goals from industry of working on actual potential therapeutics. So I chose a project in this group where I'm trying to design proteins that act as little tiny on-off switches to more specifically target cancer therapeutics to cancer cells rather than to avoid actually accidentally killing off very similar healthy cells. So you can hopefully get a more targeted therapeutic without as much of the toxic side effects that you see with most chemotherapies. So yeah, so from there, my goal is to go back into industry when I'm done with this. Yeah.
I had a quick question for you related to, I guess, industry specifically with protein design. How much of your work is computationally focused, where you're mostly just looking at protein design from a conceptual point of view, and how much of it is wet lab, where you're actually in there making things? And can someone interested in industry go either or is it something where you have a little bit of both as you're designing stuff?
Yeah, most projects in my group, and protein design is becoming more of a common thing in different companies, both as like small companies spin out of the protein design labs around the world, and as a lot of the bigger biotech companies are also picking up their own little protein design groups. Most people exist somewhere in the middle of that computational to wet lab spectrum. So I'm probably like 70 to 80% in the wet lab with just a smaller portion of computational. I know some people who are like 100% computational all the time, never touch a pipette, and they just team up with somebody else who does their experimental work for them. But it's nice to have that balance, even when there's not a pandemic, of just getting some time at a computer. But I'm the kind of person who I can't sit at a desk all day, so it's good to also still have that wet lab time.
That's pretty good. So all you, I guess, engineering students out there, if you ever get bored staring at AutoCAD and designing things in 3D for like a truck, maybe you can come on over to the protein design side and do something for biology. And I mean, now that you're with an actual protein design, are you hopeful to like go out and just join a company, or are there startups in your future? I mean, that's kind of a how would you go about doing that?
Yeah, I'm honestly not sure. I think it would kind of depend on if I come up with something new that is startup worthy. There's actually one startup that's come from our lab a couple of years ago that uses part of the project that I'm still continuing to develop in the lab now. So I would definitely consider rejoining the rest of my team who has left me for that company. Yeah, but I definitely, in my previous experience, there's definitely a very different vibe between the large biotech company and little tiny startups. I worked at both for a little while, and I definitely would put in a vote for the smaller companies, where it's really nice that you can get to know everybody who works there. You can get to know like the full breadth of a project as it moves across different teams, and just rather than feeling like a tiny cog in the big machine of this huge company, you're more part of a small community of researchers. I think my other side comment for differences between academic and industry jobs that I really miss is, at least as a grad student in academia, it's a lot more individual, where like I have my project, and yes, I collaborate on it with other people in the lab, but to get my first author paper, I need my project to be successful. And if it fails, it's really tragic for me. Whereas in industry, it's kind of nice that, like, let's say the company has like 6 different projects that are running, and if one of them succeeds, the company is successful. There's a lot more flexibility for like, if one of them isn't looking promising, even if you're like 3 or 4 years into working on it, you can just cancel that project. And it's sad, but it's less of the, I've been slaving away for most of my PhD and now my project is over, and oh no, I have to start a new project.
Yeah.
Let's forgive the word choice. Sorry, I've been working extremely hard on this project.
Okay, thank you so much for those insights, Jillian. We're going to move on to our last presenter for time's sake, and then we can ask, I guess, more protein development or design questions from you during the Q&A. Our last presenter is Ramlin Gonzalez. I worked with Ramlin at Straub Hospital and Clinic, so she is our med lab tech. She started off as a phlebotomist like I did, and then worked into where she was. So please, Ramlin, tell us how are you doing? I know we
Hi.
just woke you up. I tried to give you a little time to get your brain running before I put you on the spot.
Hi. Yeah. Hi, everybody. Thanks for being here. All these careers, I feel like I'm a combination of all that's happened with everybody. So I am a medical lab technician at Clinical Labs of Hawaii's Straub Medical Center here in Hawaii. So I like to say that I'm kind of a non-traditional medical tech in the sense that I have a cellular and molecular biology degree. So I actually didn't go to technical school for my license. I had gone to Chaminade University and had been so dead set on becoming a research scientist. So when I graduated from Chaminade, I had applied to a bunch of graduate programs and got rejected pretty much from all of them. So in the meantime, I had to work. So that's where I met Robin, and I was working as a phlebotomist for many years at Straub, and that's where I was introduced into the clinical science world. And I know KCC and UH have great clinical science programs, but I kind of wanted to be an example of other possible careers that are out there with when you have some form of science degree. I think the process that I had to go through, all you need to, what we like to call, we challenge the exam. And I did it through the AACP's Board of Certification. You need a certain amount of a certain amount of biology hours, certain amount of organic chemistry hours, biochemistry hours, and along with my job experience as a phlebotomist, I was able to challenge the exam without having to go back to technical school because, money. But what is even a tech? What do I really do in my everyday? Well, it really kind of depends on the department that I'm assigned to. Straub is a hospital as well as an outpatient clinic. So our lab is equipped with the required testings that will accommodate both of those situations. So we have a chemistry, we have a hematology, we have blood banking, urinalysis, a little bit of a microbiology, but like not really. But as a medical lab tech, I have to have an understanding for all of these subjects because my job is to analyze many, many different kinds of body fluids, mainly in the form of blood, to kind of give the doctors an idea of what is happening cellularly to these patients so that these physicians can give them the proper diagnosis and treatment. So I kind of wanted to go into some of the things that us techs are involved in the more major departments and kind of highlight how important we are in patient care. So let's say I'm a chemist that day and I'm in the chemistry department. Our machines run through chemiluminescence and immunoassays where it'll analyze the serum or the plasma of a patient's blood sample, and our machines will analyze specific analytes that will be associated with the person's physiology. So for example, one of our more critical tests is troponin. If we get a sample from the emergency room and the doctor orders a troponin, this patient probably came in saying that he's had chest pain. I know that troponin is a cardiac marker that's tested through an immunoassay to detect possible cardiac muscle injury. So we throw our samples onto the machine, but my job as a tech is to alert the ER physicians when this patient's troponin is at a critical value. So I have to take action and contact the emergency room physician and say, hey, this patient's troponin is critical. Now this kind of gives the doctors an idea that this patient could possibly be having a heart attack. Let's say I'm a blood banker that day. I have to have an understanding for antibodies and antigens. because that's how you identify your blood type. That's how you identify possible active antibodies. So as a tech, I have to have an understanding of these clinically significant antibodies so that I can safely decide what kind of blood products is compatible to give to this person, possibly bleeding person. Another one of our more critical, or not critical, but major departments is our hematology. That's kind of the department where we see more of the different kinds of body fluids like CSF or synovial or pleural fluids. So in hematology, our machines run through flow cytometry, which will then calculate and identify those cellular components of your body fluids, like your white blood cells, your red blood cells, your platelets. So let's say our machines identify something abnormal with this body fluid. It's our job as a tech to microscopically identify these abnormalities. So this is kind of where it becomes important for us to understand disease associated with what we're looking at, pretty much. Not only in hematology, but in all the departments that I mentioned earlier. So, for example, this is what I found interesting too, is our terminology is the same but different in all the departments. Like Erin used the word differential. For us, differential in the clinical setting is pretty much where we identify these white blood cells. We do a 100 white blood cell count where we identify every single cell that we look at. So let's say I'm doing a differential and I see a large number of neutrophils. I can probably assume that this patient is undergoing a bacterial infection. Or let's say I'm doing a differential for a recurring oncology patient and I see immature granulocytes. Normally I know that this shouldn't be seen in peripheral blood, but because I know this is, like, let's say it's a leukemia patient, I can safely result these findings. So that's kind of some examples that some of the things that we are constantly discussing as techs, and just like the troponin, some examples on how critical we are in patient care. And I'm honestly very happy to say that I'm in a career that utilizes a majority of my biology degree. So yeah, that's kind of what the text. I'm actually in the process of challenging to become a medical lab scientist, which my exam is coming up in January. So if you all have questions on the specifics on how I got to challenge the exam, please feel free to ask.
Yeah, it uses quite a lot of the degree. I remember when I was studying, back when I was like, yeah, I guess I'll be a med lab tech, thinking like, oh my God, why do I need to know all of this? Like, this is getting into the realm of diagnosis. I was like, I feel like this is too much. And I think a lot of people don't appreciate how much just background knowledge you need to do some of these jobs, especially med lab tech, because it's a lot of things you didn't have to, it's things that you learn in academia that aren't clinical. So they're based in academic models. And then now you're talking about a person that was like, oh yeah, that analyte that you thought was just something that ruined your experiment is actually something pretty important in the human body, and now you have to pay attention to it. But yeah, thank you so much for sharing about that. I mean, it was an interesting journey in the hospital. Definitely a great learning experience to be alongside all the doctors and nurses. I'm sure you get to work with them. They're both a blessing and a curse sometimes when you're dealing with them in the clinical setting, and that's something that, you know, I think Ram and I would both want to really stress with your students that are considering maybe being a med lab tech, is that you're very important to the mission of the hospital. You're extremely important, and you play a very background role, which I don't think it's not advertised the amount of contribution you do to the healthcare team, because literally if you're a doctor or nurse and you're doing patient care and you don't have the results from the lab, you're guessing. That used to be our saying, right? Unless you're getting it from labs, you're just guessing. And so yeah, I'm happy you're able to come here and kind of share your very odd way of getting into med lab tech, because I was doing the same thing.
We were in the same boat. Thanks,
Yeah.
Robin.
You're welcome. And so now we're going to move on to any of the questions that anyone might have for any of our panelists. I do have some prepared questions for some of them. And so if anyone does have a question, feel free to raise your hand or throw it in the chat. Tracy, since you actually interact with a lot of students and whatnot, I have a general sense of how this has affected you.
Yeah, it's basically double the craziness. Yeah, so I'm teaching right now genetics with 2 labs. And I also have 5 research students. Oh. And so it's difficult to manage. So mainly for genetics, I made lecture online. The labs, I got creative and I made them, normally the labs hold 16, but I made them alternate every other week in person. And then, they didn't have to, they can also opt out and just do something virtually at home. That was also an option. But then I also split the 8 into 2 rooms. So there's 4 in one room and 4 in the other. So me, and my TA, like a mad person, are just back and forth between the 2 labs teaching and just making sure everybody's safe. But we all have goggles and masks and paper, surgical mask on top of things, plus, lab coats and crazy amount of space, and sanitizing everything 50 times. So I feel like it's one of the safest places, if anything, just due to the biology being really precautious with safety. So, and then my research students, we have to have a COVID plan. So the students, 3 of them are new students. So I've just been training them virtually with their project, kind of with their proposal, what they're going to start next quarter. And then the 2 that I already had, they basically just have to social distance to the lab. And there's limits and spaces for when they're doing research, say in tissue culture or in other spaces. So, it's crazy. It's nuts. And it's going to continue for a while, but something you adjust for. And like I said, with teaching, it's double the work in general. I'm doing it when we're all the professors are going crazy and just want to pull their hair out just for working 12-hour days or something. But, we do it because we want our students to have the best experience they can have in the midst of the craziness, obviously.
Yeah, my mom's an elementary school teacher that retired just at the start of this. She was like, I dodged it.
Yeah, for sure. Mm-hmm.
Thank you so much. We're going to take a brief pause from this question before we move on to Jillian. And I just wanted to address a question that popped up in the chat. I think it's directed towards Ramlin. Ramlin, did you see that question pop up?
Yeah, I did.
I saw you start smiling. Is being a med tech stressful, and is the rewards that you get from that career worth everything that you have to go through?
Yes, it's stressful, but yes, it's rewarding. Let me address the stressful part first. Well, I obviously work in a hospital, so the demands are a lot more demanding, I guess. With these physicians, you get so many, where's my this result, where's my that result, but people don't realize that not only are we analyzing samples for their patient, but we're analyzing samples from not only the hospital side, but also our outpatient clinic side. And also Straub is the established stat lab for HPH. So we're getting samples from everywhere. Robin probably remembers me running around the lab all the time with my head cut off, trying to just accommodate everybody. But it's also very important.
I suppress all that.
I'm just a blur at some points. But in the technical side, let's say I'm in blood bank and I get a positive antibody. That kind of ruins your day, really. Especially now that I'm covering a night shift, we're only 2 techs, and not only do I have to manage blood bank and hematology, 1 antibody, which actually happened last night, unfortunately, but I had 1 antibody, and that made me get stuck in blood bank all day, and I feel like hematology got abandoned. But then you have to think about the patients and like these patients are coming in with some kind of stress, you know, we have to get these results out no matter what. But it's also rewarding because I kind of had a different perspective of becoming a tech because I was a phlebotomist for so long. So like a lot of the names that we see are recurring patients. And so as a phlebotomist, I did know them. I've drawn their blood. Some of them I even heard their stories. So whenever some of these like, Strab is also a big oncology clinic. So some of these patients are getting blood tests every day sometimes. And so now that I'm behind their results, I kind of start to empathize with them and like get so excited when the results get better, you know? So yeah, that's probably the most rewarding part of my career.
Okay, thank you so much, Rama. Yeah, I miss the phrase sometimes, and then I remember visiting the lab and being like, oh, that was a very silly thought. But it is fun. You build quite a community because there's a common goal that all of you are driving at, which is to survive some shifts and provide, you know, accurate results so that you can, you know, make sure that patients get cared for. So it is like it's challenging, but at least there's like there is a greater goal there that you can remind yourself of every now and then. I'm doing this for someone else. Yeah. So thank you so much. For Jillian, I'm going to try and pivot really quick because we're getting more questions. And I feel like this question sort of applies to you. Is how did you know that this was the right career path for you, or did you? And how did you actually get started? Because I feel like you're trending towards protein design so much. Like, did you know that that's what you actually want to do, or did you kind of fall into that?
I definitely completely fell into my career at every single step. Yeah, so it basically started with just liking my chem class in high school and then just like running with that with no real specific career plans, because I think somebody mentioned earlier, the like obvious ones everyone is actually aware of with a bio or chemistry degree is go to med school or go into academia and be a professor. And I had no interest in either of those because med school seemed too stressful and long-term and money. And I have no interest in teaching, despite I also come from a family of teachers. So I kind of just fell into it. It was my first job in protein purification was because that was the one professor who emailed me back when I asked if she had an opening for a summer student. My internship is the one that I happened to get when I was graduating and had no job. Just kind of fell into more and more similar careers or jobs that I enjoyed. I guess I always did kind of have that like health directed mindset. And when I started working with proteins, I just found their whole structure really cool. And when I came to grad school, I actually didn't know protein design existed yet. I just heard Dr. Baker, who's now my PI, give a couple talks, and just the idea that I could go from just like working with these native proteins where I already found their structure and was used to like looking at it in terms of like, can we fit a little molecule in here somewhere to inhibit this? I found that really cool, but just the idea that we could use computers to design one completely from scratch was just mind-boggling and sounded really exciting. So I kind of fell into that as well, but it's been really exciting and rewarding, and worked out really nicely for a job to have in the middle of a pandemic, because when our lab got shut down this summer, we all just pivoted to, all right, I guess we're going to put the wet lab stuff on hold and just do protein design all the time. And we'll test the stuff eventually when the lab opened up again.
No, that's amazing flexibility that you guys have in the institute, which is really good. I think I have a friend in your lab, Ian, and for the longest time we didn't know whether or not he was still in the state because he's on the computational. So I think he's still in Corvallis in Oregon. But yeah, it's amazing that you guys can do that. A really quick follow-up question that's completely unrelated because I'm getting DMs. We wanted to know what your cat's name is because we see it in the background.
Yeah. This is Nutmeg. She's about a year old. Our other kitten, Rainier for short, ran away a minute ago. Oh, she is being delivered back to me for her close-up. This is Rainy. She's a baby. Yeah, and then aside from plugging my cats, I'm gonna ping, there is a protein design internship. So if the world is still a disaster next summer, albeit, you know, you all are lucky and Hawaii has this more under control. There is this internship offered by Rosetta Commons, which is like the cumulative group of protein design labs around the world. And it was offered completely virtually this year. So it's a cool opportunity to still do research, even if labs are shut. So.
Okay, thank you so much for that. I guess we'll move on to the next question. And you know, Matthew Tudhill had mentioned that we actually do have a connection with Genentech, and that was who was that? I think I do remember that.
Yeah, so I just want to bring up 2 points connected to Dr. Delgado and also connected to Jillian. Even though they're far away, and maybe none of us have met them in the state of Hawaii, with Jillian was at Genentech, and Genentech actually was founded by a guy named Herbert Boyer. And Herbert Boyer was here in 1972 on a conference, and he was meeting with a colleague late at night, and they talked about the idea of moving genes. So they moved penicillinase into E. coli to make it drug resistance. And then the next step from there was to take a frog gene into E. coli. And then after that, they moved on to a human gene into E. coli, and now we have insulin. And so that all started from a late night talk at a conference here in Honolulu in 1972.
Very cool.
Probably over alcohol, but.
Oh, definitely. Yeah, they have, I believe, a statue of that chat, of them chatting over a beer in a courtyard at Genentech somewhere.
That's fascinating. Yeah. So that's one connection towards Jillian. And then on the other side with Dr. Delgado, her involvement with SACNAS. Robin, you've received a travel award from SACNAS. You've received at least one award from SACNAS in the past, and you've presented at that conference, and many of our undergraduate students have as well.
Yeah. No, SACNAS is, I mean, this year is kind of weird because everything is virtual, but then everything is weird this year. But no, it's great. Society. I mean, I'm their treasurer now at UW, and so I get to learn. Yeah, I know. For some reason, I guess I was too happy and not busy enough, and so now I'm doing the accounting for the UW chapter, and learning all these terms like in-kind donation and what a discretionary versus a non-discretionary. It's a different kind of headache, but yeah, I still am pretty active with SACNAS out here and their work. So yeah, that story is pretty amazing. Thank you for those insights. Yeah, and KCC sends, like they said, or like Leanne said, they try and send as many people as they can to SACNAS to present their work and, you know, get into academia that way. And it's a great opportunity for them to go out and meet recruiters for graduate schools or medical schools or whatever else they want to do. So if anyone's out there and you are in an area where there's a SACNAS chapter, please, you know, stop by, talk to them. It's inclusive. They want, you know, more people to participate. If you don't have a chapter and you have nothing better to do, you can always start a chapter, and then you can go through all those steps to, you know, build this organization from the ground up, which I think is a great, you know, opportunity if you have the energy to do that. Definitely a plug for SACNAS. I think it is fairly important to, I guess, highlight, especially in the sciences, that tends to be a lot more social than people think it will be. Whether you're in teaching, you're talking to students, or even if you're in research, you have to communicate things through, you know, writing grants, which is pretty important. It's a whole nother set of like writing things, which you guys have to go through to get funding, even at, you know, that city and county level, to, you know, Ramlin has to talk to nurses, doctors, patients at some point, but I think less so as a med tech. You know, as a graduate student, I feel like I talk all the time. How do you balance your work and personal life with your students and your research and Zoom and all the technical difficulties that come along with that? How do you say like 5 o'clock, stop talking to me? Well, it's 6:22 right now, so I'm very sorry about that.
Yeah, obviously I don't have boundaries right now, don't I? Just kidding. I mean, it's rough, especially right now. I mean, I started a new job, which always requires new preps and everything. So there's always like a time where you're like, okay, this is going to be a hard year, because you have to basically set up everything again, you brand new lab space again, brand new students, brand new courses, or at least teach them a little bit different. And so for me, I mean, I'm also a mother, I have a 5-year-old, you know, and a husband that I need to pay attention to as well. And so, you know, for me, a lot of it is, you know, one, being on a team with my husband to kind of tag team, you know, I'll do this part, you do this part. And, you know, especially when it's time to be off the clock, I try and be done at 5, and then spend some time with the family, especially with my son going to bed usually about 7:30. So it's like, okay, like this is family time. And then afterwards checking in with my husband and saying, hey, you know, let's talk about how things are going, and, you know, check in and spend some time with him. And then sometimes that means after that going back to work, especially because of pandemic times or over the weekends. You know, normally I don't want to work weekends, but I have to if, you know, because right now there's a pandemic. But in a typical schedule, I would have hopefully more weekend time off, and not have to grade or anything like that, doing better time management. And so, I mean, the main thing is just having that quality time. And I also, with my family, and I also, in the past, like last year, but I had to stop due to COVID. Like, I'm a singer and I take voice lessons and I sing at places and like bars and clubs and stuff like that too. So it's just like having time for myself and doing some things that I enjoy as well is, you know, in there as well. So it's just, I literally use a Google Calendar and I have everything blocked in and say, this is time for this, time for this, time for that. And then try and just cut at some point and be like, that email can wait. I do not have to answer that email today. Emails will come in all hours of the day. You don't answer them until, you know, you're back on work hours again.
Yeah, especially now. I see Leanne clapping. There's no end to how much work invades your home when we shifted over to working from home. But it is good, I think, to set a boundary and say, like, I'm stopping here and I'm not going to do it. I think Matt is a pretty rigorous believer in, once I leave this office, he's very hard to find, even though he's very close to campus. It's really hard to get to him.
So the secret to this, everybody, was don't own a phone for 10 years.
He didn't have a phone. Oh my God, it's so hard to get in contact with Matt when I was at like 9 p.m. I don't think this PCR is working. And it was like, I guess I'm just gonna go home because there's no reaching Matt.
It's funny.
But yeah, well, thank you so much, Tracy, and thank you for weighing in that on that pro tip for the future for anyone that wants to be a PI. And so I guess with these last 5 minutes, I just want to say thank you so much to all the panelists, and again to the people who collaborated with me back there at KCC to have this happen. And again, you know, please fill out the survey if you want to have more things like this. I'd love to provide it for you guys and give back to KCC. So anything that can help us make this more interesting to you, because I understand this is another Zoom meeting. So if you're going to take time out of your day and sit in on it, I'd like to make it worth your time. And I guess in these last few minutes, we'll just go down the line. And I just want to ask our panelists, you know, would you recommend the career that you chose to someone that might be interested? Like Ramlin, why would you recommend being a med tech? Briefly.
I would totally recommend being a med tech. If you've ever thought about being in the medical field without actually being physically involved with patient care, then the med tech field is probably a great field to go into. Think about going into. Science is not only restricted to being a physician or being a researcher, is what I've learned when I was going to school. Definitely other careers are out there, is what we're emphasizing in this.
Grad school definitely has its pros and cons. It's hard. It's a long slog. I highly recommend trying to sort out what you want out of grad school, potentially before committing to it, because I think that really helps to have a goal in mind, though it's obviously not necessary. And I highly recommend industry. I really enjoy it. It's another way to, you know, influence the medical field without having to actually treat patients and have that pressure on you. More behind the scenes, or, you know, yeah.
You get to do a bunch of cool stuff I have to say. So yeah, I mean, I love it. I love being able to teach the future scientists both in the lab and in the classroom, as well as medical professionals or the future criminologists and biotech and, medical technicians. I get to teach them all, and see them move on in their lives and do amazing stuff. And so that's very rewarding to be able to do that. And it's also, in some ways, it's also flexibility in the job. Like today, or like Tuesdays, I don't have class. I mean, I technically could just watch TV all day if I wanted to. Eventually, I have to get the job done when I need to get done. Maybe I might sacrifice a Saturday for that. But if I wanted to spend time with my kid all day on Tuesday, I could, because it's as long as I'm not in classes or in meetings or with my students, that's flexibility in that.
Yeah, awesome. No, awesome responses. And I guess to kind of close up, I forgot Samantha's not here. So since I did kind of work at the capacity on the other side doing whole genome sequencing and tracking enteric outbreak clusters, if you're interested in public health and you kind of see this pandemic and think, I could probably do a better job with those tax dollars, definitely, consider going into the public health field. It's good work. It's not always the easiest, mainly because it is very embedded in the bureaucracy of, it's a government job. I mean, government jobs are always going to be that way. But even though you don't have a direct connection to these people, and you're not really helping them with their care the way you would in the hospital, what you're doing for your community and for that greater area is you're preventing more patients so that they don't have to go see Ramlin, and so that hopefully the other people who are developing drugs don't have to deal with such a massive surge of people. So it's definitely the business of preventing patients from appearing. And it is an unsung sort of profession, but it is very vital. And if anything, the pandemic has taught us is that when you mismanage stuff like that, or when those safeguards are not in place, we kind of see what happens. It affects things on a massive national and global scale. And that's my 2 cents. It's really great. You do have to deal with meetings. It's a government job. You just have to deal with that. But that's all for me. Thank you so much again to our panelists. Thank you for everyone that attended. I'm glad most of you stayed for the majority of this. If you have any other questions, please, again, you can put in the chat. You can email either myself or our collaborators, or Calais, and then we can forward them to our panelists. Thank you, everyone. That's the end of our panel. And yeah, that was great. You guys did amazing. I'm glad you could all make it.